In my previous lecture, I started with bacterial anatomy and we discussed in detail the differences between the eukaryotic cell and the prokaryotic cell, which is the bacterial cell. And we also discussed the cell wall of the bacterial or the prokaryotic cell in detail. Today, I'm going to move on to the other structures which are present in a bacterial cell, and that is the next is the plasma membrane, which lies just inside the cell wall. And this plasma membrane in both the eukaryotic cell and the prokaryotic cell is a thin elastic semi-permeable membrane which is made up of phospholipid bilayer. As you can see over here, interspersed between this plasma membrane are the various integral proteins, which are both structural and functional proteins. Now, this plasma membrane has to play a very important role in both the cells, the prokaryotic cells and the eukaryotic cell. In the prokaryotic cell, the most important function is membrane transport. Now, the plasma membrane or the cytoplasmic membrane selectively allows the molecules to move in and out of the cell. So this is known as selective permeability of molecules. The molecules like food molecules and other specific molecules are going to get attached to the receptor bound proteins, and then they are either brought in the cell by active transport, in which the membrane bound these permeases and transport system like ATP is involved, or there's going to be simple diffusion where small molecules are going to move from area of high concentration to areas of low concentration. Then osmosis is also seen, where there is free movement of water in and out of the cell. The next important function is waste product elimination. Waste product elimination is that all the waste which is produced as the cell is growing or it is multiplying, then the waste product is eliminated out of the cell through again this plasma membrane. It is thrown out of the cell. Third function is secretion of extracellular enzymes. Now, the extracellular enzymes which are produced within the cytoplasm are going to get attached to the phospholipid carriers and proteins present in the plasma membrane, and then they are taken out of the cell. Outside the cell, they are going to break down the large food particles into smaller units, which are then going to be carried into the cell either by active transport or by diffusion. Then it is responsible for respiration, and this is specifically brought about by the mesosomes. And we discussed in the last lecture, mesosomes are invagination of the plasma membrane, and its basic function is to increase the surface area. So when the surface area of plasma membrane is increased, it is going to help in respiration. Reproduction is another function of, again, the mesosomes, and as the mesosomes move in, they are going to form septa in the cytoplasm, and they are going to physically separate the cytoplasm into two parts during the replication of the cell or reproduction of the cell. Then cell wall precursors, which are synthesized in the cytoplasm, they will be transported outside into the growing cell via the phospholipids and the enzymes present in the plasma membrane. And once they go out, they are going to assemble into the growing cell wall, and they are going to help in building up the cell wall. So these are the various functions of the plasma membrane. You are asked about this in your table vivas. The next structure is the flagella. Flagella, as you can see in this slide, they come out of the cell wall, and they are present and they are outside the cell. Then they are responsible for locomotion or motility of the organism. Now, all organisms are not motile. Only those organisms which have a flagella around them, they are motile. And the important function of this flagella is that it is going to bring about directional migration of the bacteria in response to a stimulus, and that is known as chemotaxis. There are different types of flagella, which we will be discussing in my next slide. But all of these, all the flagella are filamentous structures. If you look at this picture, it's a filamentous structure. Here you can see the filaments which are coming out of the cytoplasmic membrane, and these are protein in nature and they are highly antigenic, which means that antibodies would be produced against them. It has three morphological components. The first component is this, which is in form of rings. It is known as the basal body. This basal body is attached to the plasma membrane over here, as you can see in this picture. Attached to the basal body is this hook. This is the hook which is connecting the basal body with the filament that is coming out of the cell and is responsible for the movement of the bacteria. Now, the movement requires oxidative phosphorylation, and two types of movements have been observed. Either it is anti-clockwise, which is known as smooth swim, or it is clockwise and then it is known as tumbling. This is known as tumbling. Flagellar arrangement is very important. You are asked about it in your exams. You get MCQs on the flagellar arrangement. So all of you must be familiar with what the arrangement is like. Now there could be a single polar flagella, as you can see over here. This is just one flagella coming out of the bacterial cell, and this is basically seen in Vibrio cholera, and it is known as monotrichous. So monotrichous is a single polar flagella coming out of the bacterial cell. The second terminology which is used is amphitrichous. Amphitrichous is when, as you can see over here, single flagella coming out of both the ends, or from both the poles a single flagella is coming out, and this is known as amphitrichous. And then we have a tuft of this flagella at present at one end, and that is known as lophotrichous. Lophotrichous is when 3, 4 or 5 flagella are coming out from one end of the bacteria, then it is known as lophotrichous. Now mostly the bacteria are peritrichous. Peri means all around. So in this case the flagella are seen all around the cell, as you can see over here. So this is the seen in the best example of which is E. coli, a very common organism sitting in our intestines. And the last term which is used is atricate. Atricate means that there is no flagella present in the bacteria, hence the bacteria are non-motile. This slide is again showing you flagellar arrangement. This is atrichous, no flagella present, as a result of which the organism is non-motile organism. Again here, atrichous, non-motile organism. Mono means one, so there is a single polar flagellum coming out. Lopho means tuft, 3, 4 or 5 flagella coming out from one pole. Amphi means single polar flagella coming out from both the poles. From both ends, a single flagella is coming out, and the commonest is this peritrichous. This is peritrichous, where the flagella are seen all around the bacteria, and this shows that the bacteria is highly motile. Coming to flagellar movements, as I said just now, that it could be counter or anti-clockwise, or it is going to bring about clockwise movement. The anti-clockwise movement is when all the flagella rotate in an anti-clockwise manner. They are going to move in an anti-clockwise manner, and they are going to, as a result of which these flagella, they are going to get together, they bundle together, and they are going to push like this, as you can see over here. They bundle together and they push the bacteria forward, and this is known as run or smooth swim. This is anti-clockwise movement or counterclockwise movement. When the flagella are moving in an anti-clockwise manner, they just get together, bundle together, and they push the bacteria forward, and that is known as run or smooth swim. The next is clockwise direction. When it starts moving in the clockwise manner, then all the flagella which are bundled together, they are going to come apart, as you can see over here, and each flagella is going to propel separately, independently, and as a result of which the organism is going to start moving in random directions, and this is known as tumble. So tumble is when all the flagella come apart. Each of these flagella is acting independently, and the organism starts tumbling. So these are the two flagellar movement, which again you are asked about in your exams. Next are the pili or the fimbria. Now, pili, as you can see in this picture, it is a very pretty picture showing you hair-like projections, rigid hair-like projections coming out of the bacteria. They are seen all around the bacteria, and they are made of proteins which are known as pyelin. Now, these pili are basically seen in gram-negative bacteria, and two types have been identified. One are called the common pili, which are responsible for attachment of this organism to the specific receptor sites. Once they get attached, they start multiplying and they start forming colony, and they are then responsible for disease production or virulence of this organism. Then the second are the sex pili. Now the sex pili play a very important role in conjugation, and during sexual conjugation there is going to be genetic transfer of toxic genes and drug resistant genes from one bacteria to the other. So we will be discussing this sexual conjugation in detail when we do bacterial genetics. So the two common pili are the common pili and the sex pili. Next is the capsule, which is the outermost covering of some bacteria. All organisms do not produce a capsule, but there are certain organisms which are responsible for this capsule production, like the strep pneumoniae and this Klebsiella pneumoniae, two organisms responsible for pneumonia. Both pneumococci and Klebsiella are organisms responsible for pneumonia in human beings. And then there is another bacteria which is known as Bacillus anthrax, responsible for anthracosis. It is also a capsulated organism. Now, if you look at this capsule, the capsule is basically a product of metabolism, which is continuously secreted or excreted by the organism. And two types of the capsule have been identified. Mostly the capsule are polysaccharide. These polysaccharide capsules are seen mostly in the bacteria, with the exception of one organism which is anthrax. The capsule is polypeptide in nature, and this polypeptide is D-glutamyl polypeptide, and this constitutes a very important examination MCQ. Bacillus anthrax is the only organism which has polypeptide capsule, and this polypeptide capsule is D-glutamyl polypeptide. Next are the staining reactions. Capsule normally does not take up any stain. So with Indian ink technique, we are going to see the capsule, colorless capsule, and the rest of the background is picking up the color and becomes dark in color. The capsule, the bacteria has picked up the color, but the capsule remains unstained, and this is an example of negative staining. Here you can see fluorescent stain, where the organism has taken up the green fluorescent, and the capsule is giving you green fluorescent, whereas the rest of the background is again dark brown in color. What are the functions of the capsule? Very important organism which protects, very important structure which protects the organism from being phagocytosed. So it plays a very important role in the virulence or pathogenesis of the bacteria. Secondly, it is going to protect the organism from antibiotics and from being dehydrated. It is antigenic in nature, as I've told you, so that it's going to stimulate antibody response, which will help the organism, the organism will then be removed from the body. Beside capsule, slime layer is also seen around some bacteria. Now, slime layer capsule is generally very thick, as you can see over here. It is much thicker even than the diameter of the bacteria. However, slime layer are just tangled masses of polysaccharides seen around some organisms. GLYCOCALYX is a very important structure. This is also polysaccharide in nature, and it basically is secreted by many pathogenic organisms, and it is going to adhere these organisms firmly to the foreign substances which are present in our body, like catheters or like heart valves or like prosthetic joints, and they are going to form a thin film-like cover on these structures, and then obviously are going to play a very important role in the virulence or pathogenesis of these organisms. Best example is Pseudomonas, which are going to produce a very important role. This polysaccharide is going to play a very important role in cystic fibrosis, which is a respiratory tract infection, and strep mutans, which results in production of glycocalyx, and this is going to produce caries of the teeth. We will be discussing these organisms in detail and the conditions in detail when we move on to specific organisms. Next, I'm going to discuss the staining reactions with you people. Normally, the staining reactions, you're going to do the staining reactions in detail in your practical classes, and this constitutes a very important observed station during your OSPE exam. So, but I'm going to discuss a portion of it here as we are going to talk about the different staining reaction as we discuss each bacteria. All the dyes which we use for staining have been classified into two major groups. Either they are basic dyes, which are positively charged, and they are known as cations also. Example of which are the common dyes being used like crystal violet, like methylene blue, like safranine, or like carbolfuchsin, etc. And then we have acidic dyes, which are negatively charged. And best example of which is the Indian inks, which are used for staining of the capsule. The stains can be simple stain. When we talk about simple stain, that means that we are just using one stain or one dye. And this one, because we're just using one dye, the entire slide will be covered, colored as that particular stain. For example, this is methylene blue, and you can see the entire slide, all the organisms have taken up the stain, methylene blue stain, and they look like, they just look alike. Then we have different stains, and differential stain is the one in which two different stains are used. In which two different dyes or stains are used. The first stain is the primary stain, and then we are going to use another stain which is known as counter stain. For example, with this differential dyes like gram staining and Zn staining. These are two examples. Gram staining and Zn staining are two examples of differential stains which we very commonly use in the laboratory. Gram stain, one stain which we use is crystal violet, which gives blue color to the organisms, as you can see this blue or purple organisms, and the second is safranine, which is going to give pink color to the bacteria. And that is the basis of gram staining, which we'll be discussing in a few minutes. So by using differential stains, we are going to differentiate between gram positive organisms, which are purple in color, and gram negative bacteria, which are pink in color. We are also going to differentiate between the different shapes of organisms. They can be cocci, which means that they are rounded in shape, as you see over here. These are cocci, or they are bacilli, where they are in form of rods, or they can be in between cocci and bacilli, where they are known as coccobacillary forms. Thirdly, the arrangement is also seen by means of the differential stains. Either they are present in clusters, as you see over here, or they can be seen in diplococci, two cells together, or they can be seen in long chains. So the arrangement will be visible by using these differential stains. Zn stain is used for Mycobacterium tuberculosis or acid-fast bacilli. So the acid-fast bacilli with Zn staining will pick up the red stain. They become red in color, and the rest of the background of the slide remains blue in color, which is because of the methylene blue or the counter stain which we are going to use. So this is differential stain, where we are using two different stains. One is called the primary, and the second is the counter stain. Then we talk about special stains. Special stains are the ones which are used when we are trying to observe specific part of the cell, which cannot be revealed or which cannot be shown by the conventional Zn stain or by the Grams method. And example of which are the capsular stains, where we are using Indian ink technique. The capsule is not going to pick up Gram staining or Zn staining, so we are using a negative stain here, Indian ink method. The capsule remains unstained, and the rest of the slide picks up the stain. Similarly, here you can see the spore stain. The spores have picked up this red color. You can make up the red spores here, and the bacteria remains blue in color. And this is the flagellar stain, and if you look at it carefully, you can see this flagella, polar flagella coming out of the bacteria. And these structures cannot be observed by the routine staining techniques like the gram stain or the ZN stain, which I have just discussed. Then is the Indians or negative stain or the Indian ink technique, and I have told you just now that this is basically used to see the capsule around the bacteria. Now the principle behind this is that the Indian ink is negatively charged, and so is the surface of bacteria, as a result of which this stain is repelled by the negatively charged surface of the cell, and the cell is not going to be stained, but the rest of the background picks up the stain and becomes dark in color, as a result of which the capsule becomes visible. The other example of this are the stains which are negative stain is used to stain the spirochetes, which are curved bacteria, and these spirochetes we'll be discussing in detail about the spirochetes as well, and they are not picking up the stain, but the surrounding area becomes darker in color, as a result of which the organism becomes visible. Another slide showing you all the stains which we have discussed. This is a simple stain where just one color is being picked up by all the organism. All the organism has picked up one color. They are all blue in color. This is a differential stain, which this is gram staining. You can see both gram positive and gram negative bacteria over here. This is again a differential stain, which is acid fast or ZN stain, and you can see the acid fast bacilli, the red organism in a background which is blue in color. And these are special stains, which is spore stains, where you pick up the spores, they are red in color. It's also a differential stain. The organism remain blue in color, whereas the spores pick up the red color. So we have done all these in detail now. I just want you to know the different types of stains that we are being used in the laboratory, because you're asked about it in your viva. Now coming to gram staining, which is something very important. Gram staining was first done by Hans Christian Grams somewhere in 1853. He was basically working on two organisms which are responsible for pneumonia, and I've already told you the one is Klebsiella and the other is strep pneumoniae. Both are responsible for pneumonia, but Klebsiella are gram negative, whereas strep pneumoniae are gram positive. So he was the one, Hans Christian was the one who was able to first identify bacteria on the basis of gram staining, whether they are gram positive or they are gram negative in nature. It is very, very important. The stain is very important because it constitutes the first step towards bacterial classification and identification. So the first step towards a classification is whether the organisms are gram positive or they are gram negative. Now bacteria which pick up the purple stain are gram positive bacteria because of the thick peptidoglycan layer that they have. The ones which pick up pink color are gram negative. They are considered gram negative, and they do so because they have a thin peptidoglycan layer. So in other words, the gram staining, the principle of gram staining is based on the thickness of the peptidoglycan layer in a bacterial cell. The reagents which we use for gram staining are, because it is a differential stain, we are using a primary stain. We are using two stain. One is the primary stain, which is crystal violet, which is going to color all the organisms purple. They become purple in color when we use crystal violet. And then we are using iodine, which acts as a mordant, and it is going to form a crystal violet and iodine complex, which is going to get fixed to the peptidoglycan layer. So this is iodine which we are using, and it is going to fix the crystal violet iodine complex to the cell wall. And then we use decolorizing agents, acid alcohol. The function of decolorizing agent is to wash out the crystal violet iodine complex from gram negative bacteria. This is because the peptidoglycan layer in gram-negative bacteria is very thin, and when you treat it with acid alcohol or decolorizing agent, the outer membrane, which is a lipid layer, melts out, and the crystal violet iodine complex is then lost, or it comes out of the thin peptidoglycan layer. Whereas with gram-positive bacteria, because it has a very thick peptidoglycan layer, the crystal violet iodine complex remains trapped within this layer. Then we are using the second stain, which is safranine. It is the counter stain, and this counter stain is going to color these gram-negative bacteria pink, because now they have lost their dye. There is no dye present over here, so they are going to pick up the pink color or the safranine dye and hence become pink in color. Whereas the gram-positive bacteria remain purple in color. They are not affected by the counter stain. So we have used crystal violet, which is purple in color, and it is the primary stain, which is picked up initially both by gram-positive and by gram-negative bacteria. Then we are using iodine, resulting in formation of crystal violet iodine complex, and which will be fixed to the peptidoglycan layer in both gram-positive and gram-negative bacteria. But when we use decolorizing agent, which is acid alcohol, the outer membrane of the gram-negative bacteria, which is lipid in nature, it melts, as a result of which the crystal violet iodine complex is lost, and it is also because it has a thin peptidoglycan layer. Whereas with gram-positive bacteria, because it has no outer membrane and the peptidoglycan layer is very thick, it is going to trap the crystal violet iodine complex within the cell. And when you counter stain it with safranine, gram-positive bacteria are not affected, but the gram-negative bacteria are going to pick up this counter stain and it becomes pink in color. So in other words, the gram-negative bacteria are pink in color and gram-positive bacteria are purple in color. Keeping this in mind, one of your very important examination question is the principle of gram staining, and unfortunately 90% of you are not able to answer this question. What is the principle behind gram staining? The principle behind gram staining is the difference in the cell wall of gram positive and gram negative bacteria, and I have just told you in detail what the difference is. So during staining reaction, when crystal violet iodine complex is formed, it is retained by the thick peptidoglycan layer of gram-positive bacteria. And that is why they are not counterstained by safranine and they remain purple in color. Whereas the gram-negative bacteria, which has a high lipid content, and high lipid content in the cell wall. This lipid content, initially it takes up crystal violet stain, but when it is decolorized with acid alcohol, the high lipid content is going to be dissolved and the crystal violet iodine complex is removed, and this cell again becomes unstained. And this unstained gram-negative bacteria will be counter-stained with safranine. We are using safranine, which is a pink stain, so it is going to now pick up the pink stain and it becomes pink in color. So in other words, gram-negative bacteria are pink in color because of the counter stain which it has picked up, and the principle is because it has a thin peptidoglycan layer and it has outer membrane which is lipid in nature. It loses its crystal violet iodine complex, whereas the gram-positive bacteria had a thick peptidoglycan layer, so it is going to retain the crystal violet iodine complex. Now, how are you going to report a gram stain slide? Which this is again one of your examination question. You are given a slide, gram stain slide, where you are supposed to report the slide. So the first important part is you look at the slide and see whether they are gram positive or they are gram negative, or you see both gram positive and gram negative organisms. So this is a slide where you see both gram positive and gram negative bacteria, and a large number of these organisms are seen, so you can give them as 1 plus, 2 plus or 3 plus per hyper field. Then you look at the morphology of bacteria, you see whether they are cocci or they are bacilli or they are coccobacillary forms. Like here they are diplobacilli, two bacilli sitting together. Here they are again diplobacilli, and here you see clusters of gram-positive cocci. So you have to tell us about the morphology of bacteria. Then On the same slide, we are going to look for pus cells. You can see these polymorphs very well defined, 3 or 4 nuclei present in the pus cell. Number of pus cells, they may be 2 plus, 3 plus, or 4 plus. And then you look for the epithelial cells. You can see this well defined epithelial cell. This is the nucleus of the epithelial cell, and you have to give us or you have to report the presence or absence of epithelial cells. And then another very important structure are the yeast cells or candida. You see these yeast cells, these are the yeast cells, budding yeast cells, and these are the hyphae which are present on the slide. So this is how you report the slide. First you tell us whether they are gram positive or gram negative, then you have to mention the bacteria, you have to give the morphology of the bacteria, you have to mention the presence of pus cells, epithelial cells, and the yeast cells. Now, one of the important examination question is, why isn't the bacteria stained by gram method? There are certain bacteria which do not pick up gram stain. What is the reason behind it? For example, we have talked about mycobacterium or the tuberculosis. Why doesn't it? Why isn't it stained by Gram's method? It is because it has a waxy layer around it. It has too much lipid in the cell wall and the dye cannot penetrate, and that is why we are using acid fast stain. Then we talk about Treponema pallidum. We will be discussing all the organisms which are mentioned on this slide in detail while we move on to special bacteriology. So Treponema pallidum are very, very thin. They are spirochetes which cannot be seen by ordinary microscope, and you need to have a dark field illumination to see them. Mycoplasma and uroplasmas, we have already discussed, they do not have a cell wall, so they are not going to pick up the stain. And we are going to use special stains like Giemsa or Diner stain for them. Chlamydia and Rickettsia, they are both intracellular parasites, parasites which are present inside or the bacteria which are present inside the cell. Hence, obviously, they will not be seen by gram stain. So here I'm going to end. We are done with the bacterial anatomy, and I have discussed with you in detail the bacterial cell wall, which is very important. We have discussed the functions of the plasma membrane. I have done flagella with you, I have done fimbria with you, the capsule with you. So you just need to know all these structures associated with bacteria. You should know whether they are intracellular, which means that they are present within the cell, and the demarcating factor is the plasma membrane. Anything which is outside the plasma membranes are extracellular, and which are within the plasma membrane are intracellular. So the assignment for you today are 4 of these MCQs related to this topic. Please answer the MCQs with your attendance. It's hardly going to, if you have listened to my lecture, it will hardly take 2 to 3 minutes to answer the MCQs. Normally in your exams, we are asked not to use the terms like not or except. But I have given these 2 MCQ specifically with this word not and except, because there are certain examiners which are still using these words, and you get MCQs related as well. So thank you very much.